All right, I am gonna start sharing my screen. Can everybody see my screen? All right, awesome, thank you.
You're good.
So welcome everyone to this month's StaPH-B call. I just wanted to give a couple of very brief quick updates before we kind of start the call, just because I've been kind of seeing more and more members kind of trickling in, and I just think that it's fantastic the way that we have grown over the years, and how our numbers have changed and where we're at right now. So I just wanted to kind of give a little bit of a background as to, some quick updates as to where we're at right now. So there's at least 195 people on the mailing list, which I think is just absolutely fantastic. We came from this group of, ragtag group of bioinformaticians who, just kind of got together I guess it was like 5 or 6 years ago, and decided, hey, we wanted to make sure that we were all on the same page and find a way to kind of keep in contact with each other and make sure that we were staying up to date with, things that were relevant in the field. And now we have, at least 195 people who are interested in the calls, which is pretty fantastic. And of those 195 people, they make up 61 different states and organizations, which I think is absolutely incredible. So I'm really excited about that. I did want to show a map. So I made this quickly before the call. So this is our coverage as of today. This is all the emails that I have on the mailing list from all of the different people in different states and different organizations which are represented. So we still have a few states that I don't have people on here for. And so if you see a state that's adjacent to yours, or somebody who's in your region for like the BRRs or something like that, who you think would be interested in joining the calls and would benefit from this, make sure to reach out. We are always looking for more people, and we're always, really looking for people to help out with this group too, because I think that the more people we have, the more interest we're going to be able to encompass, and the more we're going to be able to, better serve the StaPH-B community. So I'm really hoping that we can get every one of these states colored in the next, hopefully in the next couple of months. So yeah, make sure to reach out with anybody who you think would benefit from this information.
Hey, Logan.
Yes.
It looks like we had in the chat there is someone from Arkansas, so we can check that out.
Oh yes, awesome.
Also,
okay, well, and so that was also what I wanted to say. If you don't see your state colored in the map, then let me know. So okay, well, I will have to go ahead and color in Arkansas. Yes, that's exciting.
Yeah, I have some contacts with the Mississippi lab, so I'll try and see if we can get someone from there.
Okay, awesome. Well, good to know. And I just put up, so this is the contact information for some of the steering committee members for StaPH-B. But I also put up here, if there are any topics that you'd like to see discussed, or any cool new pieces of software that you'd like to get more information about, or anything that you'd like to see from StaPH-B, let us know, because for the most part, I'm just kind of picking topics that I think are going to be useful to the community. And I try to be kind of mindful of, where things are going, what kind of different projects are kind of popping up in more labs, and what is going to be relevant to the community. But, we always really benefit from when people participate and they actually, make their voices known as to what pieces of information they'd like to see and what software is going to be beneficial and useful. So please don't hesitate to reach out. We are always really happy to hear from everyone. So I've got the information for the steering members up here, and so I kind of put them on blast, but they are super helpful and always really willing to hear your suggestions. And so with that, I just wanted to say thank you everyone for being a part of this, and give a warm welcome to today's speaker, Dr. Levy, who is going to be talking about Freyja. So I will go ahead and stop sharing my screen, and I will go ahead and pass it over to you.
Awesome. Thanks for the intro, Logan. Excited to be here as well. And I'll start sharing now, hopefully. All right. Okay, hopefully you can all see my slide, or first slide.
Yes.
All right, awesome. Cool. So I guess I'll take it from here. I know some of you may have seen part of this as part of a CDC Spheres call a while back. I'll tap into a bit of that and build on it today and talk a little bit more about the tool that we've developed for this analysis. So I'll go ahead and start talking. So as you all well know, this virus, SARS-CoV-2, continues to evolve. And we see new variants popping up all the time. And one thing that we need to do is be able to figure out, are they dangerous? Are they things that we really need to be creating public health advisories for, interventions, etc. And all of this data, for the most part, comes from clinical sampling, data that we're collecting from patients. There are some environmental samples as well that are being collected, but far and away, most samples that are being included in our understanding of the evolution of SARS-CoV-2 are coming from clinical sources. And that presents a pretty significant issue when you realize just how few cases are actually being sequenced and can enter into this analysis. So some countries are great about doing all of this sort of stuff. You see countries like Denmark and Iceland, different countries that sequence almost everything. They actually get tested for and test positive for SARS-CoV-2, but most countries are sequencing a very small fraction of the samples that actually test positive, and this means that we have pretty large blind spots in terms of understanding the evolution of this virus, and it is evolving around the globe. It's not just nicely evolving in Denmark where we have perfect coverage of everything. So this is something that's been studied pretty widely, like how these blind spots actually pop up, especially in our country where we're still sequencing a decent number of samples, not quite at the level of Denmark, of course, but the samples are also biased as to where we're getting this information from. So we've got issues like socioeconomic status that have been studied, where people just aren't able to access, or for whatever reason are not able to get tested as frequently in certain areas that may not have the resources, or the other option, or one other issue is proximity actually to testing. Certain areas of the country, it's just hard to get a test because you have to drive many minutes, hours to go and actually get a test done. So these are further things that are biasing our sampling, and we want to be able to do something that's going to be a little bit more all-encompassing and unbiased in this way. So as you all well know, I'm sure at this point, wastewater surveillance is a very promising alternative here. It doesn't depend on clinical sampling, and you can collect a single sample that basically is a broad representation of what's going on in the community. And for the most part, what this looks like is you get a single readout as to the level of virus that's apparent in the community. So you can see, here we're tracking concentration of virus in wastewater, and that tracks very closely, as you can see, with actual case counts. This is a plot for San Diego, where Scripps is located, and yeah, so you can see that these two kind of nicely parallel one another. And before I get too much further into this, I'd like to first, make clear that this is a pretty big collaboration between my lab and the Night Lab, as well as a few other groups in the San Diego area. In particular, this work has been a collaboration with a postdoc in the Night Lab named Smriti Karthikeyan. And yeah, so she's been the kind of experimental head on this project, whereas I've been more of the computational person. But they in the Night Lab, Smriti has been the head of that, have put together quite an incredible experiment in the San Diego area. And what this looks like is kind of twofold. We have a very a very fine-grained picture of what's going on the San Diego or UCSD, University of California, San Diego campus, where we've got some 130 odd samplers that are basically able to pick out what's going on at a particular building level. So you can see here on the left, we've got each of these little markers is a sampler, and every day it's being tested to figure out whether or not that particular building is testing positive for SARS-CoV-2. And we're able to detect even a single case in a building with very high confidence. And on the other side, we have a very kind of more coarse-grained but community-wide picture. So we also have these large community wastewater treatment facilities. Here, this one is the main one for San Diego at Point Loma, and it covers about 2.3 million people, which is the vast majority of people in the greater San Diego area. We've also recently added in a couple of additional large wastewater treatment plants that kind of fill in the last couple of gaps, but this is still 75% of the people in the San Diego area. So to first start analyzing this, we kind of use first a sort of existing approach of if we detect wastewater in campus samplers, how does that correlate with the number of clinical positives that we're seeing on campus? And again, much like others have seen, we find, yes, that the two are very correlated, and there are different effects that we see that are arguably more campus specific, but just real world, where you have people moving in and out, and that leads to huge jumps and potentially makes things a little bit more complicated than if people were living in some sort of closed system. But this is pretty close to being able to examine a closed system with these minor artifacts in move-ins and move-outs and things like that. But this is nice. We're able to get a sense as, yes, like the two track very nicely together. But when we actually sequenced the data, we saw there was a much richer picture potentially there than just looking at the actual positive or not, or concentration of virus in the water. So here's two different measures of viral diversity here on the right. You can see that wastewater is clearly much more diverse according to these two metrics. And yeah, so this suggests, well, it's not just one sequence that's in here. It's not just one type of virus. It's potentially many different viruses all mixed into one virus mixture. And how do we kind of get a handle on that? So to do that, we put together this method that's now called Freyja. And the way the method works is it starts out by basically building up a lineage barcode set. So these barcodes, there's one unique one for every individual lineage that we've now seen. And we basically encode them in this pretty simple format where basically each lineage corresponds to a single row of this. And each lineage, whether or not it has a particular mutation, is encoded as 1 or 0 in this larger this matrix. And once we've kind of done that, and the way that we do that actually is pulling out the lineage defining mutations from the global phylogenetic tree that's generated as part of Rob Lanfear's analysis and then input into Usher, which I'm sure you're all familiar with. But yeah, so this thing has thousands of lineages in it now. I mean, 1,200 I think was a number that I picked out like a month ago, so who knows where we're at now. But this is an ever expanding picture. But one cool thing is that we wanted to be able to work with everything all at once. So we're not necessarily restricting ourselves to a few lineages that we expect to be there. So this allows us to be very flexible. And the next step is we wanted to be able to basically use some existing tools for variant calling to feed in to our approach. So in our lab, we've also developed a method called IVAR in the past. We use this kind of under the hood to do variant abundance frequencies. So you can see individual SNPs, individual single nucleotide variants, here are encoded by their fraction of abundance in a particular sample. So we've basically taken in a sample from wastewater, some sort of mixture, and we now re-encode it in terms of these different abundance fractions for the different SNPs. And then we kind of bring everything all together with a mathematical approach. This is like a convex optimization approach where basically this barcode method or barcode matrix is then inserted into this linear optimization problem. And that's this matrix A, and B is the individual sample that we're looking at. So in this case, we're basically trying to solve a constrained convex optimization problem where we, enforce realistic constraints on what the sample should look like. All of the different fractions should sum to 1, and everything should be non-negative. So most things will be 0, but some things will presumably not be 0 in terms of their fraction in the sample. By things, I mean lineages, of course. And yes, we've now wrapped all of this in together as part of the Freyja model. So to test this thing out, make sure that we're not just putting together a nice looking package with nothing in it. We tested this out with a spike-in experiment here and this is in collaboration with some of our collaborators over at UCSD. We took 5 well-known lineages, VOCs, from the laboratory setting, and we mixed them in known proportions to be able to see if we could actually recover them using this computational approach, the different fractions. So we mixed them, we sequenced, and then we used Freyja to actually recover these individual fractions. And as you can see from this picture, the correspondence is quite good. We importantly, even in some of the mixtures, which go down to as low as 5% of an individual lineage versus 95% of other lineages, or just maybe one other lineage in like a 95-5 mixture, we don't see any dropout. So suggests that this approach is quite robust, even when we start decreasing the fraction of a particular lineage in the mixture. And in practice, I'll talk a little bit about this later. We've gone even lower. We've been able to identify the presence of individual lineages even below 1%, and then confirm that with PCR, VOC PCR testing for particular mutations. But that's something I'll get into a little later. And of course, we can look into individual lineages that are present in individual mixtures that we've created to see if there's any biases. And there seems to be some small little deviations depending on which mixture was in there. But for the most part, there's a very strong correspondence between our predictions and what we're expecting in these mixtures. So again, these are just mixtures, but we wanted to get a sense as to, does this work at all in like real samples? Obviously, we've got these nice things that we've created in the lab, these mixtures, but the reality is a much more gnarly picture. And to do this, we actually took out samples that we expected would be mixtures. And many of them were. And basically using the sort of qPCR VOC panel that I mentioned to you guys just before, we're able to confirm the presence of different variants that we are able to predict or to determine are actually there using the Freyja approach. So that was a nice confirmation that this is something that is probably going to be robust to real experimental data. So when we actually put this into the field, we did so in about, it was end of November of 2021. The epsilon variant was already kind of well circulating in the San Diego area. But we actually were able to detect both the alpha and delta waves before they were detected clinically, which was nice to be able to see that there's actually this ability to find things early on. And we find things on the order of 1 to 2 weeks early, generally in wastewater so far. But this was our first sort of indication that not only could we figure out a bit early that the amount of virus in the wastewater would be going up, but actually we could detect individual lineages as they emerge and rise in the community. If we zoom in further onto the UCSD campus, we see something that was actually a little bit alarming, is that actually we see huge gaps in detection of some of these VOCs, while we see them pretty consistently in wastewater. So this basically showed us that we actually had quite a lot of cryptic virus spread on campus for, in some cases, even months, that was not detected by clinical surveillance, but is continuously identified in wastewater. And of course, we can then use this sort of approach in a more general approach, not so targeted on any particular lineage. And you can see here, we see a bit of a different picture in the wastewater that we do on the clinical sampling or clinical surveillance. And that's not entirely unexpected, right? We're just discussing all these different biases that could exist. But we do see, for the most part, very similar trends as to which lineages are present in the wastewater in San Diego. So this sort of slightly different, but still similar picture was encouraging to suggest that, yeah, we can kind of do this analysis to get a sense of what the community dynamics, look like and complement clinical surveillance in a nice way. We also wanted to test to see how this would perform on more closely related variants. So, we now had, hundreds of Delta lineages. We wanted to see if we could consistently pick out certain lineages that were present in San Diego. And we were able to do this. So as you can see, alpha variant kind of decreases in San Diego in late June. And then you see a few different Delta variants arise. And this is from our campus sampling. We also saw very similar lineages in our community-wide sampling. This gives us an indication that what we're seeing on campus is actually quite similar to the larger community, and indicates that there's not like entirely separate dynamics going on in the community. The community is actually closely interacting with campus. The two are very much interconnected. And then, of course, we have the Omicron wave, which we're all intimately aware of, which we detected in San Diego on the 27th of November, this past fall. And it was not detected clinically for another week and a half or so until the 8th of December. And you can see, again, the decline of Delta. And this is all coming from our large scale Point Loma wastewater treatment facility. And one thing that we can do now, since that we're just focusing on the Point Loma facility, is we can actually scale by the actual concentration, the level of virus in the wastewater, to get a sense as to like the absolute copy number of each individual type of variant in the wastewater collection. And one thing that you can't really see as well from this picture, but you can see here, is that actually the Delta variant didn't necessarily decrease that much in its actual abundance in the San Diego area. It kind of consistently sort of just hums along, doesn't disappear whatsoever. It's just that Omicron really just took over. And it kind of makes this picture somewhat deceiving, right? That it's not necessarily totally petering out. There is still circulating Delta in San Diego. And of course, we can zoom in more on individual lineages, first of Delta, which obviously kind of decrease in abundance over time. But we also see the emergence of multiple different Omicron sublineages. First, BA.1, which emerged in San Diego. Again, it was the first type of Omicron that we found on the 27th of November. But quickly, BA1.1, which has an additional mutation at 346, takes over in the San Diego area. And only recently we've seen some evidence of BA.2, but it doesn't seem to be necessarily picking up very much. So BA1.1 is a pretty fit variant as far as we can tell. And again, we can look at the scale picture and see that many of these same lineages that were circulating in end of December continue to circulate in January, although some of them have certainly disappeared from our surveillance. Other things that we can do that are not just Freyja oriented is we can actually use some of these lineages that we've pulled out from campus wastewater. And if they're clean enough, we can actually include them in our ongoing kind of phylogenetic analysis of what's going on campus. So we can actually use this to connect individual cases to one another to say, try to inform interventions and actually have actionable epidemiological insights as to what's really going on campus. So, for example, one such case, which was a nice little case study here, is in early January of last year, we found a wastewater positive wastewater sample in kind of this northwestern region of the map. And this person was identified by UCSD campus detection protocols, and they were brought into the isolation dorm. So initially, it looks like, okay, this picture has been resolved, person is detected, they're brought to the isolation dorm, and no more SARS-CoV-2 of that particular virus is going to be found on campus. And then almost a month later, we see not too far away evidence of a very much related, I think this one's actually the same virus, in a building not too far away from the original detection, suggesting that while the original containment protocol worked okay, there is some bleed over of that virus into the community, but it didn't seem to persist for much longer than that. So we can start, trying to figure out how well our containment protocols are actually working. And this sort of information, not so much the variant lineages themselves, has been, but positive detections has been very important for the UCSD community. Individuals can go online and figure out what their current risk profile looks like by looking at each individual building on campus and whether or not it has been determined to have positive samples in the last couple of days. And this has been used to directly inform UCSD campus guidance as to masking protocols, in-person learning, etc. And also we display all of this information in real time to anyone on the internet, really, but it's meant to advise public health guidance in the San Diego area. And this is just a little video of kind of what what this looks like on the internet. So a couple of additional things is one is I've been working with some of the members of this group as well, particularly Kevin Leawood over at Freyja, to make sure that not only is this code available for people that are working with wastewater and want to do code developments, but anyone in the public health world that is interested in using this in their analysis of local wastewater streams. And this is, as I said, I've been working with folks at Freyja and CDC to make sure that this is happening. And so this is now available. There's a full workflow totally available on Terra. And again, hats off to Kevin Leawood, who recognized this stuff early and helped me bring this into the community use. One final little thing here I thought I would throw in. I guess I'm kind of speeding through this, but I guess that'll leave time for questions. So, well, one is I've implemented a bunch of new features that are now going to be, some of which are now available and some will be available soon. One is we're including methods to be able to do more error estimation to get a sense of what are the different possible confounds in our analysis that should make us believe or not believe in what we're doing. So one such method is a fast bootstrapping method in which we're able to resample the input data and get a sense as to, upon many resamplings, how much spread do we expect in the assigned lineage fractions in our sample. So here's a maybe nice looking example where we can see that this mixture is a mixture of alpha and gamma. And we see that upon many, many resamplings, the actual estimated fractions of the two are largely consistent. You can get error bounds and what that looks like. And another big question, and I'd be happy to talk more about it, but I've now recently put together some approaches for actually identifying novel lineages using a couple of different methods. Again, I can talk more about that if that's of interest, but basically using information like phasing information or other kind of statistical modeling approaches to be able to predict whether or not, not predict, I guess it's the wrong word, to be able to infer whether or not a new mutation is part of an existing lineage that is now evolved. So you can then generate a new lineage that's basically contains these additional mutations. And then an additional thing that we've been working on, and I'm not sure if this will go on to the next release, but will be available relatively soon, is being able to extend this to more general analysis of virus spread. So in particular, I've been working on Zika because there happens to be some nice data sets available for it, and it's been working quite well. With that, I'll kind of wrap this thing up. Again, want to thank Smriti and all the folks in the Knight and Anderson labs. I'm a member of the Anderson lab over at Scripps, I should mention. And as well as Peter de Hoff, who was key in getting the spike-in experiments going. So that was very much his work. And then Kevin, again, over at Theogen, who helped bring this into the public health sphere. And with that, I'd be happy to take any questions anyone's got. I realize I kind of moved through that quickly, but I have a lot of extra slides if people want to talk about, talk turkey. So
yeah, no, I think that this offers a great opportunity now to kind of ask some questions and to kind of get into a little bit more of a discussion if we want to. I know that there were a couple of questions in the chat, so I figure we can kind of go back and maybe address some of those. I think some of them were kind of talked about a little bit, but I know Dwani asked, so if there's a new lineage that is not even defined, how would that be defined, or rather picked up in wastewater?
Yeah, so depends on which version of things. In the version I'm developing, we'll have methods to actually identify new things. But in the current implementation, what it will do is it will assign that lineage to the most closely related lineage that it has in the library. So, for example, if you don't have BA1.1 yet, but you do know that BA1 is in the mixture, it will assign, sorry, take that back. If you don't have BA1.1 in your library yet, but you do have BA.1, and then BA1.1 is in your mixture, it will be assigned as BA.1 because you don't know that BA1.1 is a thing yet. Of course, now with these additional methods that I will hopefully roll out in the next couple of days, we've had a bit of a craziness over the last few days with these origins preprints that we've put out, but that's how it will be assigned, and hopefully I'll have that in the next couple of weeks.
All right, awesome. It looks like Colleen said, is there a correction for shedding post-infection where clinical samples wouldn't continue to be collected?
That's a good question. Not something that we've necessarily directly put into the analysis, but it could be included if you wanted to more potentially accurately represent the clinical picture from wastewater. That sort of kind of like a modeling approach where you actually sort of shrink the tails is an interesting approach. Something we thought a little bit about, but we haven't implemented any methods for that. It's mostly just trying to look at this empirical picture. And yes, you do see this continual like post-infection shedding, but the level of virus tends to be much lower. So shouldn't contribute as much.
Thank you. And then it looks like Karen in Texas asked, is there a built-in option for making plots that look like the bootstrap results? So like those bootstrap plots, do you have some sort of package or something in there?
Yeah, so, well, right now, the way I have it set up is the bootstrap just sort of outputs all of the pieces that you would have as part of one of these box plots. But then I allow the user to, plot them on their own. But yes, this is a good point. I could definitely have a separate option where instead of just reading out all of the values that you would place into a box plot, instead the user can just have a box plot that they don't have to make themselves. So that's something that's definitely worth adding in. Yeah, I was trying to get that into the system quickly. But yeah, would definitely be a good idea.
Yeah, no, I think that sounds great, and it's awesome that we can hopefully influence some of the kind of utility of the program.
For sure.
Looks like Molly in Colorado asked, could you discuss a little how the tiled amplicon step used during sequencing may or may not bias the relative proportions of lineages found in the sample?
Yeah, that's a good question. Let's see if I can get this to move over. Okay, so one thing that's kind of important to mention here is a couple of different aspects of the analysis that are meant to like be robust to some of these amplicon specific effects. So one is when I'm actually doing this analysis, we're using a depth weighted approach where everything is log transformed. So if one amplicon has a ton of depth, but another one has very little, you're going to trust the one that has a lot more depth, but you don't necessarily want it to be grow linearly with that. So that log transform we found has brings in a lot of robustness. One, to be able to trust the things that are better covered in your sample, but also still allow for some, again, it's log transform, so you still have a lot of weight associated to things that may be lesser coverage in the sample. And the other thing that's really important is, well, so one is if you have complete dropout, then you're still going to have pretty strong ability to predict a lineage as long as it's not just that one mutation that you unluckily happen to miss. Basically, if something is not in a particular sample, it gets down weighted all the way down to 0. And then it uses, that basically doesn't factor into the error calculation at all anymore. And you can use all the other mutations that are present in the sample and in the known lineage to be able to make your prediction. Also, the approach is pretty robust to just things that don't agree with your actual barcodes. So one thing that I think is a little bit informative as to why I use this one norm here is if you had an example problem where you had one lineage that has these 4 mutations or not, and then a secondary lineage that looks like this, and you have a sample that has 100% of each of those mutations. If you solve this problem in a 2-norm scenario, you actually get that it's a mixture of the 2, even though it's totally incoherent with the 2. And of course, if I expand this much longer, the fractions, so imagine instead that I had 1s going all the way down and then everything was 0 here, you're still going to get a fraction of this second lineage showing up in your sample. And it's all, in proportion to the amount of error in each different component. So you get here, there's 3 versus 1, so you get, a 3-to-1 sort of ratio here. But if you solve the problem using a 1 norm, you actually get the thing that is most kind of there, most likely gets 100% of the weighting. So that sort of relationship has allowed us to be very robust to all the things that are in the library. And we've got well over 1,000 different possible lineages, so we need to be able to set things to 0 when that's the case. And there's a long explanation, mathematical explanation as to what that looks like. I can get into if people are interested, but it's similar reasoning for why why like a lasso sort of approach works. So in this case, we're doing, kind of shrinkage in the actual error space rather than applying like a regularization penalty. But yes, it's robust in that way.
I'm trying to see now. Okay, it looks like Dwani had another question.
Sure.
So let's say Omicron was not defined yet and we start seeing it in wastewater. How would we know something new is surging? And I was actually wondering about that too. Like if you don't have that,
Yeah.
if you have something that's completely novel, is there like a way that you could ID something as like not defined yet?
Yeah. So I alluded a little bit to some of this stuff, but I wasn't sure how much time I would have today, but apparently I flew through this much faster than I expected. So I've put together kind of two approaches for doing this. The first of which is using actual like read level information to say, I know I have this existing lineage, but then, in a place where I know there's a particular mutation, there is another unique mutation that shows up on that read. So at that level, you have a pretty good sense, assuming, things aren't too noisy in your reads, that if you're seeing repeated instances of one SNP on a single read, and then that another SNP somewhere on the same read, you can start to guess that, yeah, something new is popping up. And if it happens with sufficient frequency, you can start saying, okay, this thing is a new lineage that's present in our sample. And yeah, is closely related to lineages, whatever that in your sample. And again, since we're starting with such a broad library, even something like Omicron, which we don't have a lot of the intermediate virus lineages, you can still start building up these, piece by piece. The other approach is, well, we're working with amplicon sequencing, so a lot of the time actually being able to like reconstruct a full haplotype is not going to work because we don't have unique SNPs and overlaps or things like that. So you can use instead sort of a more mathematically driven approach where you can add in individual lineages by basically making perturbations to your existing lineage set. So you basically create a new lineage, call it A tilde, with it's basically one of your lineages in your set with a one mutation perturbation. And basically by iterating this procedure, you can continually drive down your error. So to like paint a picture of what that looks like, if you have particular lineage barcode set, call that A, but it doesn't yet have this new lineage BA1.1, you can then basically create a new lineage BA.1 that includes the mutation that you think might be in there. And if it drives down the error inconsistently, so I haven't included here, but in order to make this work in general, we're basically using many samples from the wastewater over time to see if this is something that's robust, especially with an emerging lineage where it's going to be coming in a relatively small fraction. You want to make sure that this thing is really there. So you can basically sum these problems over multiple days in order to get a sense as to whether or not this lineage is really popping up, or if it was a sequencing error, contamination, whatever, that made it look like something was in there. But this allows you to attempt to infer whether or not a new lineage is in there, even if you don't have the phasing information, the read level information to be able to connect the two directly.
Thank you for the explanation.
Sure.
All right.
Yeah.
I can open up the floor now to anybody else. I think we got to all the questions in the chat. But yeah, if anybody has any questions that they would like to ask, go for it. I've got a couple here, but I'll let people kind of ask first before I jump in.
Sounds good.
Does anybody have any burning questions? So in the chat, good stuff. I have to imagine that read length greatly influences these calculations.
Yeah, well, so of course, if you have really small little amplicons, you're not going to get much out of phasing level information. Of course, if you have very long ones, I mean, if you did like, that's another thing that I guess I didn't really touch on. If you had like really long, like nanopore style reads, you're going to have way more noise. And I have found in some examples that, well, it limited test set, but I found that the Freyja approach does work well on nanopore data. But yeah, so of course this is only going to work, so well if you have tiny little amplicons. But larger amplicons, you'll be able to use the phasing information more readily, for sure.
If you did have kind of noisier reads and maybe you're using something that wasn't as accurate, like it had a little bit of a lower like Q score when you're sequencing, is that going to really affect this, or does that kind of come out in a wash when you do your kind of like log transformations and all?
Yeah, in general, I mean, right, it depends just how bad it gets. Right, if it's truly like really poor quality, then it's hard to really make many inferences. But in general, even with like, I mean, for example, just to think about like a data set that we've been working with, in many cases, I've seen coverage values go, 50, 60% region, and we can still reproducibly estimate the lineages that are present in the population. Like samples from one day and then the next, one has great coverage, the other one doesn't, but you still get the same sort of lineages appearing even in the low coverage case. Of course, if you get unlucky and all of the coverage that you need is in all of the wrong places, then it doesn't really help even if you have like a ton of coverage. But in practice, we've seen that you can do pretty well even with full dropout of mutations that are part of your lineage dividing set.
So it sounds like the wet lab method could kind of influence some of this as well. Have you found that there are certain either primer sets or methods that have worked better or worse with this?
Yeah, so, well, I mean, the primer sets are kind of the same issues that we've seen, I think, for clinical samples, where you see like a huge dropout in particular regions. So as long as, for example, for Omicron, as long as you're keeping with the most recent Arctic protocols, etc., it seems like that's fine. In terms of like the wastewater side of things, the folks in the night lab they make their protocol publicly available. I think there's added a couple of little modifications here and there since, but they've, even since when we started this experiment, have totally improved things to like by miles. So now early on we were still getting pretty good coverage, but like we had a lot more samples where we would just see like nothing. Now almost all of the samples come back with 99+% coverage, even in wastewater. Of course, this is there's just a lot more virus around right now, but even in sometimes in the preceding period, where there was a lot of infections going on, we were still not seeing that kind of level of recovery. So it's pretty cool to see that that's going on.
Nice. I have a question that kind of takes a hard right turn, I feel like, but I figure if people don't have, oh, Kevin, it sounds like you have a question.
Hey, not so much a question, but did want to make a note on, Josh throwing out that we made these workflows available on Terra. Also wanted to stress how helpful it was that Josh made this available through a GitHub repository. Often you see these tools, you see this kind of functionality. It's like, how the heck do you access these things? Making it available through a version controlled repository made it easy, especially through a Conda environment, in a Bioconda environment, the way that Josh did, made it super straightforward for us to containerize it and then capture it into a little workflow that a lot of public health laboratories are using. So for any lab that is interested in getting access to this, through that AMD agreement we have to support SARS-CoV-2 analysis, there is both the GCP support to set up a Terra workspace, and then also just the contact support from Fusion folks. If laboratories want the walkthrough on how to import this workflow, what are the required inputs, and even help for doing the Freyja plotting and visualization there. So, and again, huge thanks to Josh for being responsive to a lot of the feature requests that we get from public health laboratories that I funnel to Josh, and making those available on the workflow itself. So that's just what I wanted to say. So thanks, Josh. Thanks, Logan. And yeah, if anyone has questions there, feel free to reach out to us.
Yeah, definitely. All of the suggestions, I've been getting a ton of suggestions, via personal communication, or just, people that bring up issues, pull requests, whatever, on the GitHub page. That's been super helpful, and not just, making the method, a little bit more streamlined, but also just, having more of the features that people really need. So it's one thing for me to make a package that works for me to put out a paper, but it's, been very helpful to hear exactly, what is useful to everybody else that's using it for actual public health uses.
That is really excellent. And we really do appreciate, you being so responsive, and for working so well in the kind of public health. We're kind of constrained in a lot of ways in what we're able to use, and what, kind of our experience level is. And so I think that this has been a tremendous help for a lot of people. So I guess before we move on away, I did want to ask my question, which is a little bit different. So this tool has pretty much, it looks like, been used for wastewater kind of prediction, and in terms of the percentages of lineages that are available. But could it also be useful as a tool for kind of contamination checks? I know we've seen, in some cases there have been, the delta crons and things like that are popping up. And so is this a tool that you could use to potentially kind of mitigate some of that misinformation?
This is something that we've done a little bit of work on, for sure. The delta crons, even some of the stuff that I've been involved in some of the SARS-CoV-2 origins, where, highly contaminated early samples come up, and we're trying to figure out, what's really going on. Is this really an Antarctic soil sample, or what's going on? We're able to use it in scenarios like that to figure out, this is actually a combination of these two really early lineages, or whatever. We've also been able to pinpoint not so obvious contamination, for sure, where we see, delta appearing either way before it should, because some samples weren't processed until much later, or way after it should, because there's continued contamination with some delta lineage virus in the lab. So it's a quick way of us to be able to say, okay, well, this is, early pandemic, but the sample didn't get analyzed until later, and, it's 20% delta. Well, there's something going on there. That's definitely something we've been using to identify things that need to be resequenced or reanalyzed.
Josh, hey, this is Joanna here. So I figured I should speak up once at least. I've been asking all these questions. Thank you for this amazing tool. It's been useful, and we have actually used it to look for contamination. So I actually did have a related question for you. I know we're calling it at a full genome level. Is there a way to kind of just stratify and look through, especially maybe to figure out recombinants as well? Can we do a scan across the genome to see, in this portion, it's 40-60 delta and Omicron, because that's what it looks like. Or maybe this is where it looks like delta in first half of the genome, and the second half looks like more Omicron. So maybe look through the sliding window across the genome to be able to look for something like that.
Yeah, that's an interesting point. In principle, that should be readily possible, because the way that the barcodes are set up, you could basically create a different set of barcodes, where you basically restrict to individual regions. So basically, instead of using the whole genome like what we currently do, you then basically take the existing barcode set, which now there's functionality built, so you can actually pull that out into your local repository, or your local folder, and see what that really looks like. pretty easily. And then you basically, if you know, pull that into pandas, pull that into whatever sort of data frame analysis software you prefer to use, and just cut out any mutations that are outside of the region of interest and see what it predicts for you. That would work. It's not something that I've ever attempted, to be honest, but it's an interesting idea for sure.
And how often do you update the underlying library of mutations or these barcodes?
Yeah, so
especially now with all these, now even Omicron sublineages have started popping up with just one mutation between them.
Yeah, that's a really good question. So actually, initially I was using more of an ad hoc approach where I was pulling lots of genomes and whatnot from GISAID and trying to do this myself. Now I've basically, because we parse directly from the global phylogenetic tree from the Usher repository, we basically directly pull in from what is live. So I don't have to necessarily compute anything myself. You're getting up-to-date lineages that are updated by the Usher team every day. So it's all happening in real time, and it's not something you as the user have to run, or I even as the as a user have to run. This is pulled directly from Usher, and all of the lineage names that correspond, whether or not a new lineage corresponds to a particular VOC or some existing constellation. That's all pulled from another project I'm part of, the outbreak.info metadata there. So that's it's all updated in real time by people that are curating this data.
Thank you. Yeah, useful to know.
Sure.
I just wanted to jump in here and say that it is the top of the hour, and so I want to be respectful of people's time if you have to get off. I just wanted to let you know, but if you do want to stay on for a few more minutes, if there's any other questions or anything like that, I can kind of like go over a little bit. I don't know.
That works for me.
If anybody else wants to, but yeah, I just wanted to say that I'm going to go ahead and stop the recording.